It's like 5 degrees out.
I got practice at 3:30, so we got 3:40.
Aren't you guys inside?
Yeah.
Yeah. You're doing bunt defense and that sort of thing. Oh, yeah.
We haven't done that yet, so we'll see. We're getting live, so.
That machine you guys have, that machine you guys have, somebody told me that machine will throw high 80s, low 90s.
Oh, easily. The new one that we just got, can probably throw 95 to 97.
Whoa.
It's got the filthiest slider we've ever seen. I mean, it's just, yeah, it's impossible.
That'll teach you how to hit.
I think we had it going at, I think, 82 miles an hour slider, but then we moved it up to 54 feet speed. So it was like equivalent to an 89, 90 mile an hour slider.
Man.
And let's just say not a lot of people play.
Aggressive, huh?
Once you stop talking about your body starts to not quite as this one. Yeah. Not quite so anymore.
See?
One that you guys are across the street. Right? And the one that we're trying to, the other direction more towards we're 60 meters. So
Does it have a swimming pool?
it doesn't have a test in that there.
Yeah.
So one of us is home frequently.
Yeah, that's true. That's not for most people. I would imagine dogs. That's a weird scene.
Outdoor Jordan. Outdoor Jordan doesn't look. Are you guys thinking about moving?
We have to, our rent's gonna go up like $200.
Oh, what's that?
Yeah, we already paid too much, so it's gonna be like $1,600.
Welcome to Ann Arbor. Now please go home.
Where's the house?
Target Kroger, it's like behind it. That direction, it's like-
Yeah,
It's Varden.
Aldi.
Varden Drive.
Yeah.
It's like right in the Varden Street.
You're close to the freeway too. You hop right on the freeway and how's the snow coming in later? Okay. No, it's not like this guy who had to basically spend 4 hours digging his car.
That was why I did that last night.
It is fun. I'm like, do you want me to crash it, guys?
Yeah. That's the amount of- Always so efficient.
Yes.
Yeah. Kibar.
Okay, guide, read these over. Just don't go on autopilot and say, oh, I know what this is. Read them, because I might have changed something.
That to me before.
I will continue to do it too. So you'll read the bloody question. Both, supposed to be both.
Oh, interesting.
Auto check changes the model. Let me do day without auto.
Yeah, better works.
Wait, I'm sorry.
No, you saw without a correct, you have to go down it.
She?
I don't know why you made it. All the quizzes in? Anyone have a quiz out? No? Okay. All right. We were talking about enzymes, and we referred to the fact that the clear majority of enzymes are proteins, so they're polymers of amino acids. There are some enzymes, however, that have additional chemical groups that help them do their jobs. Those are called prosthetic groups. Prosthetic groups. Prosthetic groups assist in helping the enzyme catalyze a reaction. In many cases, they will hold the molecule in place, or they may accept or dispense electrons or something like that. So this prosthetic group will combine with the enzyme, and this complex of the enzyme with the prosthetic group, that is referred to as a holoenzyme. The enzyme without the prosthetic group is referred to as an apoenzyme. And the APO enzyme is not active until it gets its prosthetic group. And then when it has this prosthetic group, now it can actually catalyze the reaction it's meant to. Some prosthetic groups that enzymes contain consist of these small organic molecules, and they're referred to. Oh, okay, that's really quite interesting. What is going on there? It's off. Oh, I didn't shut you off. Why did you turn? Anyways, I was saying some of these prosthetic groups are small organic molecules, and they are called coenzymes. Coenzymes. So these are prosthetic groups that are small organic molecules. Elena, when I say organic molecule, what makes a molecule an organic molecule? Not sure? Oh, okay. Jamie? Right, contains carbon. It contains carbon. Strange. Let's try this now. For those of you on Zoom, we are having some kind of computer issues, and all right. Come on, buddy. Work with me. There we go. There we go. There, much better. All right, so the prosthetic group, cofactor, coenzyme, they are small organic molecules. So the line between them is sometimes a little blurred. There are some enzymes that will have organic molecules that actually have metals embedded in them. They're still called coenzymes. You could call them prosthetic groups. Really, coenzymes are a type of prosthetic group that consists of small organic molecules. So these, like the enzyme, they're not changed. Ultimately, they might be changed during the reaction, but they get changed back to their original form. They play pretty vital, pretty integral roles in the job of the enzyme. They may serve as electron carriers or carriers of methyl groups or something like that. They play lots of different interesting roles. So one particular coenzyme is this one called NAD, which stands for nicotinamide adenine dinucleotide, which we're just going to abbreviate NAD because I like easy. And this is an electron acceptor. So this particular coenzyme plays a role in oxidation and reduction reactions. This particular coenzyme can accept 2 electrons, and it accepts a hydrogen ion, and it donates those electrons to something else, reducing it, and it then becomes NAD again. Notice NAD without the 2 electrons and the hydrogen ion is charged. It has a positive charge. So here is the structure of NAD. You'll notice the nicotinamide ring on top there is derived from dietary niacin. Dietary niacin. So dietary niacin is required to make this particular cofactor. If you don't have enough dietary niacin, you don't have enough NAD, and you have troubles with oxidation reduction reactions, which are quite important for making energy.
My question.
Oh, we'll get to that. Another cofactor, another coenzyme involved in oxidation reduction reactions is flaving adenosine dinucleotide, or FAD. And flavine, that should remind you of another dietary vitamin, riboflavin. So this particular molecule has this flavin group up top there, and it's derived from dietary riboflavin. This is also involved in oxidation reduction reactions. And there are many other coenzymes involved in specific types of reactions. So for example, pyridoxal phosphate is derived from dietary vitamin B6. Is that better? That looked a little glary to me. Is that, yeah, better? Huh? Huh? Too much. Can you see that better now? The dark screen with these lights here, look, to me, it looked pretty glary, look hard to see. That is better, yes. Okay, then we'll keep it that way. Yeah. Pyridoxal phosphate accepts amino groups, so it is used in transamination reactions. So reactions that involve amino acids will usually involve enzymes that have a pyridoxal phosphate coenzyme. Without vitamin B6, you have trouble with amino acid metabolism, and you have trouble with protein metabolism in general. Tetrahydrofolate is derived from dietary folic acid. Tetrahydrofolate plays a role in single carbon transfer reactions. Single carbon transfer reactions are very important for the biosynthesis of many different types of molecules. And without it, you have trouble making DNA, you have trouble making RNA, you have trouble making several other things as well. Then there's biotin. Biotin is involved in carboxylation reactions. Carboxylation reactions are really important for biosynthesis and also for lipid catabolism. So here are your coenzymes. Here are the related vitamin. The one I didn't mention is this one, the deoxydenacylcobalamin. That's derived from dietary B12. This is very important in rearrangement reactions. Rearrangement reactions. Vitamin B12. Vitamin B12 is very hard for people who've had bariatric surgery to absorb. So there are commercial versions of B12 that you take under your tongue. The technology is a quick melt technology. You put it under your tongue, it dissolves very quickly. And that's how people who've had bariatric surgery can get enough B12, because it's very common for people who've had bariatric surgery to have B12 deficiencies. Some people don't absorb B12 very well, even though they might have their whole intestine. So this type of quick melt technology might help them get enough B12. I was chatting with Beth Ulrich. Apparently she doesn't absorb B12 all that well. So she will take preparations of B12 to help her get the B12 she needs. And without those supplements, she suffers, for example, she gets anemia and stuff like that. And she gets pain in her tongue and pain in her legs and what have you, which is symptoms of anemia. So certain deficiencies for these vitamins that help your body make these coenzymes have pretty significant pathological consequences. So if you don't get enough B12, you have trouble making heme. And you have something called megaloblastic anemia. Your blood cells are bigger than they should be, and they're not as dense, and they're very fragile. So because they bust open as they circulate around your body, you suffer from something called pernicious anemia. That can be very hard on your spleen. Your spleen can actually start to enlarge, and it may get damaged. B12 deficiencies are also a problem for people who follow a vegan diet, unless you are a smart vegan rather than a not so smart vegan. If you talk to Evan's dad, Mitch, he's a vegan and he's actually a smart vegan. He knows ways to get B12 from certain foods. And the Journal of Nutritional Science had a survey of vegan communities, and the rate of pernicious anemia in them was very high because they don't get enough B12. Dietary deficiency for vitamin B6, for pyridoxal phosphate, will cause this condition called dermatitis acrodynia. This is characterized by swelling of the skin. So your skin has trouble attaching to the dermis. Lesions on the hand, ears, nose, and face, because those areas that are under high shear forces, or you have to replace the skin often, doesn't get replaced that easily because you have trouble making protein because you don't have normal amino acid metabolism. Dermatitis acrodynia. Pellagra, if any of you are fans of John Steinbeck, the great American writer who wrote some of the most depressing novels I've ever read, he talks in his novel, The Grapes of Wrath, which takes place in Bakersfield, California, which is near where I grew up. It's about an hour and 20 minutes from where I grew up in the Central Valley. He talks about pellagra. Maybe you read Grapes of Wrath? Okay, depressing, isn't it? Yeah, depressing. Virtually all of Steinbeck's stuff is depressing. But pellagra is the result essentially of a niacin deficiency. In dogs, you have a disease called black tongue, where the tongue will turn kind of a coppery reddish color that looks kind of black. So pellagra is characterized by the 3 Ds: dermatitis, diarrhea, and dementia. Turns out that NAD is also important for metabolism in the brain as well. Finally, thymine deficiency will cause a central nervous system issue that will result in paralysis, coma, and then death. This is called beriberi, named after the South Pacific tribe where it was first identified, because these people ate a diet of polished rice that was low in thymine. Enzymes, of course, are very heavily regulated, particularly enzymes involved in biosynthesis. Biosynthetic pathways, energetically speaking, are typically very, very demanding. Making molecules is very expensive, energetically speaking. If you don't need that thing you're making, you shut that pathway down, save energy. It's kind of like you when you're studying. If you know your whatever, history, if you know your history really well, you don't need to spend much time studying it. You should be studying your 404, right? Come on, massage my ego. So if you don't spend a lot of time on that, because you already know about the Civil War, and you know about the Spanish War of Succession, and you know about piracy in the early 1800s, early 1700s, and so on and so forth. You're savvy on that. You know that. You don't have to put your time on that. You spend your time doing something else, studying physics or statistics or calculus or whatever. So are you done, Kelsey? Oh, you're crying about physics. Okay, thank you. Okay, you can have a moment there. Okay, that's enough. All right. So the end product of those biosynthetic pathways will tend to shut off the earlier part of the biosynthetic pathway. And this is called either end product or feedback inhibition. End product or feedback inhibition. And you can see this essentially where some sort of end product inhibits the enzyme that catalyzes the first step in its biosynthesis. That way you don't waste any of the energy required to make any of these other intermediates as well. This is extremely common. If you look at bacteria or fungi or fish or frogs or freds, this occurs in virtually all the living organisms. This is really, really common, and it helps organisms essentially devote precious resources to where they are needed and shut down pathways that are not needed. Likewise, if you have a mutation in the structural gene that encodes that enzyme, then you will not be able to make that particular end product, and you might have some sort of deficiency. And there are various inborn errors of metabolism, as it's called. So there are some inborn errors of amino acid metabolism. Some of these are involved in proper degradation of these amino acids. So phenylalanine is degraded to the amino acid tyrosine, which is then ultimately degraded to acetyl coenzyme A and then some other intermediates as well. The inability to properly degrade phenylalanine will cause a buildup of phenylalanine. Phenylalanine will cross the blood-brain barrier and it will disrupt neurotransmitter biosynthesis. So that can cause intellectual disability, epilepsy, things like that. Albinism is an inability to make melanin because of an enzyme that converts tyrosine into melanin, an enzyme called tyrosinase. Maple syrup urine disease is due to an inability to break down branch chain amino acids. That produces alpha keto acids that are excreted in the urine and import to the urine a kind of syrupy sort of constituency. And because those can get into the blood-brain barrier, they can inhibit processes in the brain, they cause early coma and death. And then homocystinuria, where you're unable to break down homocysteine, which is a breakdown product of the amino acids methionine and cysteine as well. Inborn errors of metabolism when it comes to carbohydrates as well. You can have problems with properly degrading glycogen or abnormalities in glycogen accumulation or something like that. Inability to break down lactose. That's actually due to a regulatory mutation that prevents the enzyme that degrades lactose, lactase or beta-glucosidase, from being expressed later on in your adult years. There are defects in lipid metabolism where you can't break down complex lipids. They accumulate and they can cause pretty severe organ damage depending on the organ and what have you. These are all due to defects in various enzymes involved in these pathways. Evan?
Do you want us to know the specific chart?
Yes, but not in gory detail. Enzymes are also regulated by phosphorylation. When you put phosphates on amino acids, those amino acids become quite bulky. They also become charged. That will cause the protein to change conformation. This can either activate, inactivate, or modify the activity of various enzymes. You could change its specificity for certain substrates. You can activate the enzyme or shut it down. This is a very common way of regulating enzyme activity. So the example here is in muscle cells. The hormone epinephrine, which is released by the adrenal glands and also by some sympathetic nerves. Sympathetic nerves will usually release norepinephrine, but epinephrine is released too. This will bind to a receptor in muscle cells that will cause muscle cells to phosphorylate their enzyme glycogen phosphorylase. When glycogen phosphorylase is phosphorylated, it becomes active. And it will then degrade glycogen to glucose 1-phosphate. That's how your muscles get fast energy when you exercise to the point where muscles start to become anaerobic. You get that fast energy because you're breaking down muscle glycogen. If you need fast energy, let's say Evan's on second base, he sees the ball go by the catcher, he gets that adrenaline rush, he sprints to third base faster than he ever thought he could ever run. That's that adrenaline rush. You get that fast energy. So all of a sudden, coach says, I didn't know your 40 was that fast, because he saw the ball skirt past the catcher. He wasn't expecting that, but he takes off. And he takes their base, and now he's big time in scoring position, which means that they better not swing. There are other cases too, in liver cells, for example, where glucagon will not only activate glycogen phosphorylase, it will phosphorylate, cause phosphorylation of the enzyme that makes glycogen synthase. Phosphorylation of glycogen synthase actually turns it off. So phosphorylation can turn some enzymes on, it can turn others off. So phosphorylation is a powerful way to regulate enzymes. So chemical reactions that occur inside cells are still governed by the descriptions that you read in chemistry and physics textbooks. They don't violate the laws of thermodynamics because they occur in living systems. They still obey those same descriptions. Why then don't those reactions just grind to a halt? Because of a constant input in energy. Once you have a loss of energy input, those reactions largely grind to a halt. And of course, if we have an organism that is at equilibrium with its environment, we have an organism that is dead, essentially. So this constant input of energy, that's how you keep the living organism going. So there in chemistry is this term we use called the Gibbs free energy that describes the energetics of a biochemical reaction. If the Gibbs free energy of a reaction is positive, that means the reaction, thermodynamically speaking, is not spontaneous. If the Gibbs free energy of a reaction is negative, that means that that reaction, thermodynamically speaking, is spontaneous. So positive delta G, it's not spontaneous. And negative delta G, it is spontaneous. Gibbs free energy can also be described as the sum of changes in heat, otherwise known as enthalpy, and the change in entropy, or the change in order, increase in disorder, something like that. And so this is what we were talking about before, where if your delta G is negative, the reaction will go in the forward direction spontaneously. Doesn't say how fast it will happen, it just says that it can happen without input of more energy. And if your delta G is greater than 0, the reaction will not proceed spontaneously. You need to pump it up energetically in order for it to go. So there are lots of reactions in cells that fall into this category. How do you get them to go? The answer is through reaction coupling. You take two reactions, one that's favorable, one that's not favorable, you link them together, and the one that's favorable essentially pulls the unfavorable reaction along. Think of your car, think of it on the road, think of it when it dies. It won't go. Its delta G is positive. The tow truck comes along, hoists it up, and goes. The reaction that goes, the tow truck, pulls the reaction that doesn't go, your car, along. That's the way this works in the cell through reaction coupling. So you have the conversion of A to B, not favorable. You have the conversion of C to D, that is favorable. You link it together, you couple them. Now, the conversion of A to B to C to D, that's now favorable. And your book uses an analogy kind of like this. And I think this analogy is potentially quite powerful, where you take one reaction that is not very favorable, you couple it to one that is. Think of it as this kind of cog on a wheel with a set of gears. This gear turns, that is automatically going to turn this way. That's what we mean by reaction coupling. So what's the reaction we normally use that's favorable in cells? The hydrolysis of ATP. That's the one we use. ATP, when it's hydrolyzed, releases bucket loads of energy. The bucket loads of energy can be used to pull non-favorable reactions along. So if you look at ATP and you put on your chemistry glasses, you sweat, I'm sure some of you are too. You look at this molecule, you see 3 negatively charged oxygens lined up together these three oxygens don't like each other. They repel each other. So when you add water across this bond here, you release an inorganic phosphate, you've relieved a good deal of electrostatic repulsion. That's one of the reasons why you release a good deal of energy. You are also taking a molecule that is ordered, you're making it less ordered. You're introducing more parts. That also favors the reaction entropically speaking. So if ATP is the energy currency, the energy money, if you will, in the cell ATP, then how do you make it? You take energetic molecules and you break them down to more basic substances. The energy and the bonds of those molecules are used to make ATP. That's the short version. So your energy production is going to come from the breakdown of three different energy intensive molecules. Carbohydrates, proteins, and then lipids. Each of these will be broken down, and the energy and their bonds will be used to make ATP. So proteins will be degraded to amino acids. Lipids will be degraded to free fatty acids. And then glycerol. Carbohydrates, if they're complex carbohydrates, will be degraded to monosaccharides. Start with these first. So sugars go through a chemical pathway called glycolysis. This pathway is anaerobic, doesn't require oxygen. It's a step-by-step process. Your book has all these steps in glycolysis. I'm not going to require you to know those steps. It is a long pathway. It's a complicated pathway. My biochemistry students need to be able to draw out each reaction in the pathway. You don't have to do that. Here's what you have to know. What goes in, what comes out. All you need to know. Okay? You don't need to know about glucose is phosphorylated by ATP. That's to make glucose 6-phosphate. That's catalyzed by hexokinase, and then glucose 6-phosphate is rearranged by hexoisomerase to make fructose 6-phosphate, which is then phosphorylated by ATP and phosphofructokinase to make fructose 1,6-bisphosphate, which is then broken apart by aldolase into dihydroxyacetone phosphate and glyceraldehyde 3-phosphate, which is blah, 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 and all that. No, you don't need to know all that. Okay. See, don't need to see that. Come back to it's okay. It's okay. Okay. Some of you are starting to. No, need to know what goes in. Glucose. Glucose goes in. Then you need to know what comes out. What comes out is 2. So glucose is a 6-carbon molecule. You get 2 molecules that are 3 carbons long. So you haven't lost any carbons yet. And those 3 carbon molecules are called pyruvic acid. You get 2 of those. Then you get 2 reduced NADs. Can you see this all right back there? You're squinting. No. That, can you see that? It's not better. Okay. Let's try that again. 2 pyruvic acids. Better? Okay, 2 reduced NADs, and then you get 2 ATPs out of this as well. That's what comes out. The ATP is made by generating high energy intermediates that have a phosphate to carbon bond. The energy from that phosphate to carbon bond is transferred to ADP to make ATP. That process of making ATP is called substrate level phosphorylation. Now, if all your ATP is made by substrate level phosphorylation, then you as an organism carry out a fermentation. So, if your coach makes you guys run 800s, then you rest a little bit and you run another 800, and then you rest even shorter period of time, you run another 800. Some of you are really starting to look white now because you've done that lots and it hurts. Your lungs are burning. Your lungs are burning because you can't get enough oxygen to your muscles. Your muscles are making lactate. That's what causes your muscles to cramp. Your muscles are carrying out a lactic acid fermentation because all the ATP they're making without oxygen is through substrate level phosphorylation. And when yeast or something like that have a metabolism that's fermentative, all their ATP is made by substrate level phosphorylation. That's why they tend to grow more slowly. All right, you with me so far? So carbohydrates, they go through glycolysis. Yeah.
You got about two good choices.
Picky. Just going with white. Not as aesthetically pleasing, perhaps, but hey, it works. You can read it. Now, if the cell has oxygen and it has the right machinery, it will make the lion's share of its ATP using a process that requires intact membranes called oxidative phosphorylation. That's the process by which we get most of our ATP. And this is the reason why we need to keep breathing, because oxidative phosphorylation requires a terminal electron acceptor. In our case, that terminal electron acceptor is oxygen. There are some organisms that grow without oxygen that still do oxidative phosphorylation. They just don't use oxygen as their terminal electron acceptor. They use nitrate, they use sulfate, they use something else. But we use oxygen. That makes us obligate aerobes. I think having the lights off too make it harder to see if I use a lighter color. That's, you know, all this great color chalk up here. Try and use it, but not if you can't see it. Now, inside cells, glycolysis occurs in the cytosol. It occurs in the cytosol. The rest of the metabolism occurs in the mitochondria. So the pyruvates will get transported into the mitochondria. And everything else we're going to talk about occurs in the mitochondria. Well, almost everything else we'll talk about occurs. Most of everything else we'll talk about occurs in the mitochondria. And here's glycolysis again. I just need you to know what goes in, what comes out. What goes in? Gotcha. Glucose. Gregory. The two organic acids are coming out. Very good. Jimmy, what else? Okay. And Dane. 2 ATP. In the mitochondria, the pyruvate is oxidized and is converted to acetate. It's oxidized and converted to acetate. It encounters this gargantuan enzymatic complex called the pyruvate dehydrogenase complex. It's huge, absolutely huge. You break mitochondria open and you look at it in the electron microscope, you see these granules, and those are the PDH. You can see them in the electron microscope. They're that big. They look a little bit like a pinwheel, like the kind you got at the carnival. That's what they kind of look like as a pinwheel. This acetate is carried, it's attached to a carrier called coenzyme A. Coenzyme A does a lot of carrying things in the cell. Coenzyme A is made from a vitamin called pentathene. Pentathene, if any of you are into reading the back of milk cartons while you're eating your cereal, you might notice that it says that the milk is fortified with vitamin D and pantothenic acid. No, you're not into reading milk cartons? Okay. Yeah, I still read cereal boxes and milk cartons, and yeah, see the kids who are missing, and yeah, that's on. I shouldn't make fun of that. That'd be terrible, missing your kids. From here, the acetate is going to enter a cyclical biochemical pathway that's either called the Krebs cycle, the citric acid cycle, or the tricarboxylic acid cycle. They're all the same thing. So it's called those three. In fact, your book uses those three names pretty interchangeably. They're all the same thing. So, you can call me Ray, or you can call me Gray, or you call me Sunny, or you call me Raymond. You can call it any one of those things. It's the same thing. The acetate is oxidized completely to carbon dioxide and water. So you've broken down the glucose now to carbon dioxide and water. But the Krebs cycle makes a whole lot of reducing equivalents. For every acetate, you need 3 NADHs, 1 reduced flavine, 1 GTP, and 2 molecules of carbon dioxide. So what goes in? 1 acetyl coenzyme A. The coenzyme A is recycled. Coenzyme A is just a carrier. What comes out? 3 NADH, 1 reduced flavine, 1 GTP, 2 molecules of CO2. Now, each glucose is going to give you 2 acetates. 2 acetates. So how many of these per Krebs cycle are you going to get per glucose? You're going to get 6 NADHs, 2 FADH2s, 2 GTPs, and 4 molecules of CO2. The other CO2 came from the PDH complex reaction. You gave off 2 CO2s and you gained yet another set of reduced NADHs. So now, you see that? Yeah, okay. So then it goes to the PDH complex. And from that, you get 1, no, you have 2 pyruvates, so you get 2. You get 2. And you get 2 carbon dioxides. Then it goes to the Krebs cycle. Let's just say Kansas City Krebs cycle. And you get per turn of the Krebs cycle 3. One reduced flavine. 2 CO2s and 1 GTP. And folks, GTP is equivalent energetically to ATP. They're essentially interchangeable. You have all this reducing equivalent. So glycolysis net, it generates 4 ATP, but you use 2 of them. So you only net makes 2 ATP and you have 2 NADHs. So if we take a look kind of at this whole thing, from glycolysis, we go from C6 to 2 C3s. We give up 2 CO2s with the PDH complex, we gain 2 NADHs. We get 2 acetates, we put it in the Krebs cycle, we get 2 CO2s. And there's 2 of those. So 1, 2, 3, 4, 5, 6, we've accounted for all 6 carbons. So per sugar molecule, if we do our math right, that means we get 2 plus 2, that's 4, plus 3 times 2 is 6, that's 10. We get 10 NADHs. We get 4 molecules of ATP, 2 here, 2 here, 2 reduced flavings, 6 molecules of CO2. And that's from the oxidation of 1 glucose molecule. So you're burning a little bit of ATP in order to get more ATP for work. You're investing, if you will. Now, you have a problem. Problem is this. You have oxidized glucose. All your carriers are now reduced. If you want to oxidize more glucose, you can't, because your carriers are all reduced. So you need to oxidize those carriers. Here's what's going to help you out. You're going to give those reducing equivalents, those electrons, to an electron transport chain. They will reoxidize these carriers so that you can oxidize more glucose. The electron transport chain takes the electrons and passes them to different carriers all the way down to molecular oxygen. When the molecular oxygen receives the electrons, it takes the electrons, also takes hydrogen ions, and it makes water. Water is the most innocuous end product you could ask for in a metabolic pathway. In fact, water is beneficial. However, the electron transport chain has a few tricks up its sleeve. Those tricks up its sleeve go like this. Now, this electron transport chain is embedded in the inner mitochondrial membrane. Like that. It's embedded right in that membrane. And this chain alternates between carriers that carry hydrogen ions and electrons and carriers that just carry electrons. So you have the flavine up there. It's going to give its electrons to the ubiquinone, the coenzyme Q10. But then the coenzyme Q10, which carries both protons and electrons, gives its electrons to a cytochrome. Cytochromes just carry electrons. What do you do with those hydrogen ions? You get rid of them. But you're going to extrude them on the other side of the membrane. So as the electrons go down the electron transport chain, you're extruding protons to the other side of the membrane. Go through this, electrons are getting pumped on the other side of the membrane. And remember, that membrane is impermeable to protons. Protons are sitting out there, they just can't come back in. You're now generating a concentration, pH, and charge gradient across that membrane. You're generating a giant battery. What can you do with batteries? You can do work. What work are you going to do? You can use it to make ATP. That's the work you're going to do. So these hydrogen ions are going to use this enzyme called the ATP synthase. The ATP synthase is a remarkable molecular machine. It actually turns like a carousel while it does this. It lets the protons in. As it does, the energy that is available with that proton coming in is used to make ATP from ADP and phosphate. Protons come in, makes an ATP. Protons come in, makes an ATP. Protons come in, makes an ATP. And this process whereby you make ATP through a proton gradient generated by an electron transport chain, that is the process of oxidative phosphorylation. We also call this process cellular respiration. All right, any part of that you want me to go over again? You've probably heard this before, but it doesn't hurt to hear it again.
What was that PDH complex?
That is soluble inside the matrix. It's not membrane bound, it's soluble. The enzymes of the Krebs cycle are also soluble, with the exception of one enzyme, succinate dehydrogenase, that is attached to the membrane. Because it makes flaving, reduced flaving, it sends those electrons straight to the electron transport chain. There's a part of the electron transport chain called complex 2, by which it directly communicates with the Krebs cycle. There's probably a lot more detail than you know, but that was free. All right, questions? The person who postulated this hypothesis for ATP synthesis was a guy named Peter Mitchell. Peter Mitchell was at MIT for years. I saw Peter Mitchell give a seminar. The guy was a prototypical nerd. He had a bad haircut. He wore these thick glasses. He even had a pocket protector, and he walked around like this, scooped over like this. And his first slide was all calculus. And by then, I think by the end of his seminar, half the crowd was asleep. No one understood his seminars because it was all biophysics, it was all calculus and stuff like that. The guy, in terms of mental energy, he was amazing. But oh, the guy was such a nerd. He was, oh my gosh, he was big time nerd, which means I got on with him just great. But he truly was one of the most brilliant people I have ever met. He's remarkable. Most of this he essentially did just through sheer calculation and assembling work that other people had done. He made theories that other people could test. This theory he came up with and people started testing, he turned out to be right in just about every aspect of it. Just incredible, incredible piece of biology. That's why it's sometimes you'll read in books, it's called the Mitchell hypothesis after Peter Mitchell, the ultimate nerd. So the electrons will go down the electron transport chain, transfer down the electron transport chain. It's thermodynamically spontaneous because you start out with carriers that have a low affinity for electrons. You then get to carriers at the bottom that have very high affinity for electrons. That makes the transfer thermodynamically spontaneous. As it goes through these carriers, the carriers extrude electrons because they alternate between those that carry protons, those that don't. And then the protons are readmitted into the matrix through the ATP synthase. And that synthesizes ATP. And so something essentially that the cell needs to dump, this excess reductant, is used to make large amounts of ATP. It's just really an ingenious system. And so this coenzyme Q10, ubiquinone, that acts as kind of a ferry between these different elements of the electron transport chain. You can buy coenzyme Q10 as a supplement. When my sister was doing a lot of racing, she would take that supplement all the time. She said every time she took it, it helped her race better. She did marathons and she doesn't do marathons now, her name is a shot. But she does a lot of cycling. She still takes it, says it works really well. I don't know if any of you've tried experimenting with Q10. You can get it now in a gummy solution. You don't have to swallow a pill, you can chew a gummy. So before a race, you can eat some gummies and have Q10. And so this will get you really a lot of ATP. And instead of the paltry 2 ATP for glycolysis that you got, you end up with something on the order of about 30 ATP, essentially per glucose molecule. So this is quite the investment, essentially. Now, we've essentially laid out the land with the oxidation of glucose. Turns out the oxidation of either amino acids or lipids pretty much feeds into many of these pathways. If we talk about lipids, free fatty acids are oxidized, at least in animal cells, inside the mitochondrial matrix using a process called beta oxidation. Beta oxidation requires a cofactor called carnitine. Carnitine is taken by weightlifters. You've seen it in some pre-workouts, will have carnitine in it. Evan, help me out here. What's a pre-workout that comes in a can that just about everybody in the gym I've seen uses? Yeah. C4. That has taurine and it has carnitine, and that is used essentially for oxidizing lipids in the mitochondria. You oxidize the lipids to acetylcoenzyme A. And the acetylcoenzyme A just gets fed into the Krebs cycle. So you just have one extra process, beta oxidation, and then you shove them right through the Krebs cycle, and you make 3 NAD, 1 FAD, H2, 1 CO2, and 1 GTP, and just like you did with that. And so essentially, your fatty acids are oxidized 2 carbons at a time. And if you have an odd numbered fatty acid, the 3-carbon molecule is converted to a 4-carbon molecule and then shunted right back into the Krebs cycle. So fatty acids essentially go into the Krebs cycle pretty much. Amino acids will enter after they have their amino groups taken off by transaminases that use pyridoxal phosphate as a cofactor derived from dietary vitamin B6. Their alpha amino acids either go into glycolysis or they feed into the Krebs cycle. And so they are treated essentially the same way. Now, glycolysis, with 3 exceptions, 3 different reactions, glycolysis can actually be run in reverse. There are 3 reactions that are not reversible. You have bypass reactions, and glycolysis can actually be used to make sugar in your body. So let's say, for example, let's pick on Kelsey because she's so fun to pick on. Let's say Kelsey, let's say you pitch the last of a doubleheader, and let's say you kick Iwoo's butt. Okay. In fact, you 4-hit them. They only get 4 hits off you. Okay. They score maybe 1 run, and that was lucky because somebody's throw just kind of went awry or something like that. So you beat the stuffing out of them. You beat them 6-1. Okay. So you got to reward yourself, right? So you go to Texas Roadhouse. You have a steak. Okay. You have a prime rib? No, New York? Ribeye? Sirloin. Okay. You have a sirloin. All right. Because you worked hard. You earned that, right? Okay. What, softball players? Can I? Yeah. Okay. Okay, you can have a steak too, because you're part of the game too. But all right, so Kelsey is now getting amino acids, but she just worked hard pitching. So her glucose levels are down, but the brain needs glucose. So to get those glucose levels back up, she's eating amino acids. That's okay. The liver can take those alpha amino acids, those alpha keto acids that are made from the deamination of amino acids, they can put them into glycolysis, run it backwards, and make sugar. That's how Kelsey's blood sugar level stays about the same, even though she's eating meat. That's called gluconeogenesis. Gluconeogenesis is how essentially you make sugar from small carbon precursors, usually amino acids. We'll talk more about that when we get into hormones because this is all regulated by hormones. All right, we'll finish this on Monday. We'll get into transport and ionic changes across membranes, and then how neurons essentially make action potentials.
I didn't want to get my